So we should continue now with our third speaker. We move on to Alzheimer research, and the speaker, Charles Jennings, you may know him from former times. He worked for many years for Nature as an editor. Then he established founding editor of Nature Neuroscience. So he calls himself a scientific administrator these days. So he provides actually perfect research conditions for people here at Brigham Health Hospital. And so we're really happy to have you here. Thank you. Please.
Great. All right. See if we can get it correct first time. Did I get it right first time? Everybody, can you hear me okay at the back? Okay. Wonderful. Good. Well, thank you, Rainer. Thank you to the organizers of this wonderful meeting, Oksana, Conchi, everybody else has been involved. It's really been terrific. And thank you most of all to Nikos yourself for this invitation. I'm really, really honored to be here. I thought I would just start with a brief anecdote. Nikos, you might remember this. As Rainer mentioned, I worked for Nature for many years, and I first met Nikos, in fact, via Francis Crick. When I had just joined Nature, Crick came to London doing a book tour to promote his new book on the neural basis of consciousness, the astonishing hypothesis. I went to his lecture, I bought the book, I stood in line and got Francis Crick to sign it. It wasn't really an opportunity for a long conversation, but I took the book home and I read it, and there somewhere in the middle was a little section on the work of this guy I'd never heard of, Nikos Logothetis, who was doing this fascinating work on bistable perception, training monkeys to look at ambiguous stimuli and report what they saw and record their neural responses. And I thought, well, that sounds pretty cool. A few months later, this manuscript landed on my desk. I was now the editor at Nature in charge of neuroscience, and so I thought, this is fantastic. So we sent it out for review. It got good reviews, and we accepted it for publication. But it was a fairly complicated manuscript. It was a fairly difficult reading if you were not a systems neuroscientist. So we thought, we need to make this a little clearer to a general audience, and who should we ask to write a commentary on it? And so we knew that Crick was interested in this area, and we asked him, and he agreed. And so he wrote this News and Views, and I think it made a great package, and hopefully really helped to raise broad interest in the question, the challenge of understanding the neural basis of perception and consciousness. As Rainer said, I then went on to launch Nature Neuroscience, and soon after we launched, I got another manuscript from Nikos, and this one, I remember vividly, you were calling me, if not every day, at least every week, to see whether the reviews were in yet and how it was going. And this, of course, was the first paper on bold fMRI in monkeys, and indeed the reviews came back. They were very favorable, and we were thrilled to publish it. And I went back to PubMed before this meeting and looked up Logothetis plus fMRI, and there's now 175 papers, of which our Nature Neuroscience paper was the very first. So I just wanted to pass that on and say it's just been an honor to have played a small part in this wonderful history of accomplishment, Nikos' career, and much more to come, I have no doubt. So back to my title. So, the brain's sorrowful mysteries, the challenge of developing treatments for neurological disease. Those of you who had a Catholic upbringing might recognize the reference to the mysteries of the Rosary. So these are meditations on the life of Jesus Christ. And so we have the joyful mysteries illustrated here. We have glorious mysteries, the Ascension over here. But in between, we have the sorrowful mysteries around the Passion and the Crucifixion. We've heard in this symposium on the mystery of the brain, we've talked a lot about the glory of scientific accomplishment and the joy of celebrating the accomplishments of our friends and colleagues. We've said less, maybe perhaps until this morning, we've said less about the sorrowful mysteries, the idea that the brain is not only the source of joy, but also the source of suffering, and brain diseases as a source of great misery and problem in the world. This is statistics from the World Health Organization looking at disease burden, and the point here is that of their top 10 disease categories, 5 of them are brain-related, including depression, cerebrovascular, so stroke, Alzheimer's and dementia, and many others. So collectively, these are a huge societal problem. I'm going to be focusing specifically on Alzheimer's and dementia for a couple of reasons. One, it's a huge problem. It's among the biggest problems, I think, in clinical neuroscience. It's a disease for which we have no effective treatment at all at the moment. There are treatments available. They are purely symptomatic, and I'm told by my clinical colleagues that they are largely and only marginally effective at best. I myself am not an expert on Alzheimer's, so I will be primarily channeling the expertise of some of my colleagues at Brigham and Women's Hospital who are experts. And so I am very much a student in this field, but I hope I can pass on to you some of what I've learned. So primarily Dennis Selkoe, who is the director of the Romney Center for Neurologic Disease, Tracy Young-Pierce and Dominic Walsh, faculty members at the Romney Center. In fact, Dominic recently moved to Biogen, where he is now heading the Alzheimer's translational research effort. And then Risa Sperling, who is the head of the Center for Alzheimer's Research and Treatment at Brigham, and I'll be talking about her work on clinical trials for Alzheimer's. So, quick outline. I'm going to start with a bit of background on Alzheimer's, which will, I suspect, be familiar to some of you, but perhaps not all of you, particularly those working in systems neuroscience. I'll talk briefly about disease mechanisms, particularly talking about the amyloid hypothesis and the genetic and other support for that idea. I will talk about efforts to discover new treatments, including a bit of recent work from the Brigham, and then we'll end with some discussion of clinical trials, including a trial that's being led by Risa Sperling. And I think I hope it will give you some sense of the challenge of getting from scientific understanding to actual effective treatments. It is an enormous challenge. Just get some water here. So this may be familiar to many of you, particularly those of you who are local. Alzheimer's disease was discovered by Alois Alzheimer, who first described it in 1906. And in fact, his first presentation on Alzheimer's, which I actually given here in Tübingen, I'm told that the hall is fairly close to where we are now. So his patient, Augusta Dieter, she was a lady who in her late 40s started to develop dementia, delusions, loss of memory. She became very distressed at times and apparently would start screaming, and eventually she was committed to the asylum in Frankfurt, where she became a patient of Alzheimer. She died in 1906, and he obtained her brain and examined it under the microscope and described the pathologies that we now recognize as the classic hallmarks of Alzheimer's disease. So this is a more recent patient. This was actually a patient of Dennis Selkoe, who died quite a few years ago. But it shows the two hallmark pathologies. This large orange blob, this is an amyloid plaque. and then you see it surrounded by these dark objects here, which are neurofibrillary tangles. The plaque is made primarily of, as we now know, of a short peptide called amyloid beta protein, or A-beta for short. And these tangles here are composed of phosphorylated tau, which is a protein that is associated with microtubules. And it becomes deposited in neurons. Eventually, the neurons die, and they leave this sort of ghostly outline of tau protein. The pathology spreads gradually through the brain, and eventually, in advanced Alzheimer's, you'll see this very substantial degeneration. So I think you don't need any theory of dynamical systems theory or fancy neuroscience to recognize that there's something very seriously wrong with this brain. Clinically, Alzheimer's disease is a slow, insidious, progressive disease. It is distinct from the sort of cognitive decline that I think all of us, or maybe all of us except Nikos, experience as we grow older. I mean, I'm constantly forgetting where I left my cell phone or which level I parked my car. That is not Alzheimer's disease, at least I hope not. Alzheimer's is distinct. It starts with this sort of preclinical phase in which cognitive deficits are very subtle, goes on to what we now call mild cognitive impairment, a prodromal form of Alzheimer's disease. It progresses to full-blown dementia, in which people are impaired in memory, they become confused, they become disorganized, they have increasing problems with the tasks of daily life. And then in its final phase, they become physically impaired, they'll often be bedridden, often very apathetic, and eventually can develop problems with feeding, with drinking, and will die of the complications of Alzheimer's if they don't die of something else beforehand. So it's a very serious and very unpleasant disease. The major risk factor, of course, is age. And it becomes much more prevalent with increasing age, particularly starting in your mid-60s or 70s, such that by the time a person is 90, they have roughly a 40% chance of having clinical Alzheimer's disease. This is a big problem given the aging of our populations. So this is global. World population. Along the horizontal axis is numbers, vertical axis is age from birth to 100 years old. This blue triangle in the middle represents the world population in 1950. So lots and lots of young people, lots of babies, very few old people. Actually, I think if you look carefully, I think that spot, is that Nikos, that little dot right there, 1950, is that right? I'm somewhere there as well, just a little bit above. So that was the population 1950. We're now 2018, so that's roughly where we are now. Very different. So the population has increased about threefold, but more importantly here, it's also become much older. And that trend is going to continue according to demographers. So by the time we get to 2100 or even 2050, the middle of this century, we're going to have many more old people relative to the number of young people. And unfortunately, many of them are going to develop Alzheimer's disease. This was really brought home to me, sort of the human cost, from some news stories that I've seen from Japan, and it just happens Japan is further along that curve than most other countries, and so they now have it's a serious problem of dementia in Japan. I saw this news story was talking about the problem of demented people who just go missing. They get confused, they get lost, and the government is now collecting statistics on this, and apparently it's now about 16,000 police reports a year of demented people who've gone missing. Most of them are found within a few days, but about 500 of them die, typically road traffic accidents. They walk in front of a truck or something. So sort of brings home the human cost. And many neighborhoods are now organizing search groups. This is a group, they're doing search and rescue for missing person alert. So that's Japan now, but that is the future to which we are all heading. if we don't come up with something for the disease. You might say, well, what can we do now? Are there interventions right now? Some evidence, this is a very recent report from the US National Academies, some evidence that cognitive training is helpful, will reduce your risk, blood pressure management, physical activity, staying mentally alert, mentally active. These are things that we probably try to do anyway, but they are at best only going to have a marginal impact relative to the coming demographic tidal wave that we're facing. So we need new treatments, and they are going to have to be mechanism-based treatments based on the scientific understanding of the disease. So what do we know about the course of the disease? So, as I mentioned, the most prominent feature of Alzheimer's pathologically is these amyloid plaques. They are made of a short peptide, which is called A-beta, amyloid beta. It's either 40, the predominant form is either 40 or 42 amino acids, shown right here. A-beta, this red peptide, is produced from a longer precursor protein called amyloid precursor protein, or APP for short. And it is released by the action of two proteases. So one is called beta secretase, which cuts right here, and another called gamma secretase, which cuts down here. And the sequential action of these two proteases releases these short peptides. They are soluble, they're released into the brain, they form dimers, trimers, and larger oligomeric complexes, which are soluble and which are not fully, these are not fully known. Even though the peptide sequence is described, the exact conformation of these things is still not exactly known. It's a mixture, and that's actually likely to be critically important for understanding the disease mechanism. The oligomers go on to form these larger fibrils. They then deposit into plaques that eventually solidify into these amyloid plaques that we see. Just to highlight what we think of as the culprit. This is the APP protein. It was first discovered, in fact, because it's on chromosome 21, which is the chromosome that is triple copy in people with Down syndrome, and we know that people with Downs are at very high risk of developing Alzheimer's disease, and that was the clue that led to the discovery of the protein. And this will summarize an enormous amount of work over a decade or more. But a critical discoveries made in the 1990s were rare, highly penetrant genetic forms of early onset Alzheimer's disease. And these mutations, the first mutations, were in this APP protein. And a number of such mutations have been discovered. And very importantly, they cluster around the sites of those two proteases. So the beta secretase site here, the gamma cleavage site here, which is actually within the membrane. APP is a transmembrane protein, and then other sites that are actually within the released peptide itself. So these are highly penetrant mutations. If you get one of these mutations, you will almost certainly develop Alzheimer's disease in middle age. Another gene was discovered called presenilin-1, which also has highly penetrant mutations that, again, give you early onset Alzheimer's. And a key discovery was that the presenilin-1 gene actually encodes the catalytic subunit of the gamma secretase. So we now have very strong evidence that the release of this peptide, A-beta, is critically important in driving the disease. So we know that the mutations in the APP gene tend to increase the rate at which it is cut by the gamma secretase, and the mutations in presenilin-1 also affect the processing of APP, tend to accelerate the release of this pathological peptide. Interesting point here is that some researchers here in, I think in Munich, were able to find Alzheimer's original slides a few years ago, and were able to extract, including the Augusta Dieter, and they were able to extract DNA, PCR it, and confirm that she in fact had a presenilin 1 mutation. So we now know why she developed the disease. Another very important discovery came in 2012 through genomic studies in Iceland, and through sequencing of several thousand Icelanders genes, they discovered a very rare mutation in APP, which has the opposite effect. This is a protective mutation, which reduces your risk of developing Alzheimer's. Unfortunately, it's a very rare mutation. It's about, I think, less than 1%, even in Scandinavian populations. There's probably nobody in this room who has one, unless you happen to be from Iceland. But it turns out that this mutation actually slows down the cleavage, the action of beta-secretase. And so this is really important because it addresses a point that Bob alluded to earlier, the relationship between these highly penetrant monogenic forms of the disease and the more common late onset form, and this happens in Alzheimer's as well. The fact that you have a mutation in the same gene that is lowering your risk of the late onset form strongly implies that the two forms are the same. I mean, they look very similar pathologically, and this strongly implies that they have a shared underlying causal mechanism. An interesting point that's maybe not been as publicized as it might be, is that this gene not only protects you against Alzheimer's, it also seems to be protective for normal, healthy cognitive aging. So apparently people who your chance of reaching the age of 85 without any significant cognitive impairment is increased by a factor of 8 if you carry this gene. So that implies that the process that's driving Alzheimer's disease may also, in some slower and more benign form, may also be driving normal aging in what would otherwise be regarded as healthy people. Quick summary, so the genetic landscape as a whole, this is a survey from 2013. I don't think the picture has changed dramatically since then. So up in the top left, we have these highly penetrant mutations in APP, or the presenilins. Very strong effect, but they are very rare, so they account for only a very small percentage of Alzheimer's cases. More common is ApoE, so this is a protein that is involved in transport of lipids and cholesterol, and one particular variant of it called E4 is present at a frequency of, I think, about 14%, and that substantially increases your risk of Alzheimer's. It's not a certainty in the way it is with these rare forms, but it's in population terms, this is the biggest genetic driver of Alzheimer's risk. Another one that was, and I should say we don't know exactly how it works, although we do know, and I'll show a little bit of data to indicate that it increases the production of the A-beta peptide by a mechanism that's not fully understood yet. Another important gene is called TREM2, and this is a receptor on microglial cells, so these are immune cells within the brain, and it's thought that they have some function in clearing debris, and again, we don't know exactly how it works, but it suggests that immune mechanisms may also be important in determining the course of the disease. And then there's a large number of more common genetic variants that individually have much less effect. This list will have grown since, it has grown since 2013, but the story is basically the same, and a number of these genes are involved in the immune system in one way or another. So again, that reinforces the idea that the body's immune response to the disease process is somehow important in determining your risk. So that's the genetics. Knowing what the genes are is great, but it doesn't tell you how they act in this slow time course. until a few years ago, the only way to make a definitive diagnosis of Alzheimer's was through post-mortem. So the patient would have dementia during life, but then you would see the plaques and tangles post-mortem. It's now become possible to make a definitive diagnosis during life thanks to the development of PET ligands. This is positron emission tomography. So these are radioactive ligands that bind the amyloid plaque, and you can image them non-invasively in live patients. And so here, CN, this is a cognitively normal subject with very little deposit of amyloid. This is an Alzheimer's patient who has a very substantial deposit. And this is somebody who is still cognitively normal, but is already showing some buildup of amyloid and are quite likely to go on to develop clinical symptoms. So what about the time course? So this is equivalent to the diagram, the chart I showed you earlier, showing the increase in disease risk with age. This is showing this green curve is the increase in the frequency of the pathology. So the dark dots were with post-mortem histology. The green are using the PET non-invasive imaging, and so it's gratifying to see that they co-align, suggests that the PET is indeed a reliable indicator. Now, you could look at this two ways. You could say either, oh my gosh, you know, there's all these people walking around, they have Alzheimer's and they don't know it yet. Or you could take the glass half full view and say this means that we have 15 years, right? There's a 15-year window of opportunity in which we could potentially intervene to prevent the or slow down the progression from having amyloid in your brain to developing clinical symptoms. So that will be the hope. This just shows, this is from the Harvard Aging Brain Study, so this is a prospective observational study being led by Risa Sperling at Brigham and her husband Keith Johnson at Mass General Hospital. So this is, the blue are people who do not have amyloid in their brains as seen by the PET imaging. Red are those who do. They're following them following individuals over a period of 5 years, and they're measuring PAC as a composite cognitive score. So what you see is the blue people do fine. In fact, they get better just because they're coming back every year and doing the same tests, so they get better with practice. The people who have, the people in red who already have amyloid, they decline on average each year. So I should emphasize these people were cognitively normal. They did not have a clinical diagnosis at the time that they were scanned or enrolled in the study. So the beta amyloid accumulation in the brain is predictive of cognitive decline even in people who don't yet have clinical symptoms. And then on the right, these are just some subcomponents of the overall score. Just won't go through this other than just the key points. So firstly, there are many more women than men affected by Alzheimer's disease. That is primarily because women tend to live longer than men, and there's some debate as to whether the risk is higher if you normalize for age. What does seem to be the case, though, is that once you've accumulated A beta, as seen by PET, women seem to decline faster than men. This is based on pooling of several cohorts, including the Harvard Aging Brain Study. And this also shows that, I won't go through the details, but it shows that ApoE4 also affects increases the rate of decline. I mentioned two pathologies, two hallmark pathologies, A beta, and the other one is the deposition of tau. And so within the last few years, people have also developed PET ligands for tau imaging. And so you can scan people for both. So here again is a cognitively normal individual with only a little bit of amyloid and a very small amount of tau. This is somebody who's cognitively normal but has a strong amyloid deposition, and now you can see the tau is appearing. So it starts in the entorhinal cortex, it spreads to the inferior temporal lobe, and then gradually spreads throughout the brain. And if we understood better how that spread occurred, that might be a promising target for intervention. And then this is a patient with full-blown dementia, and they have both amyloid and tau widespread throughout the brain. Tau levels are also, again, in people who are initially healthy, tau levels seen by PET are also predictive of decline. And in particular, this is if it's in the hippocampus or the inferior temporal cortex. What you're seeing, so to the right are the people with higher tau, and this is the rate at which they decline over a 2-year period. So the take-home from this slide is simply that high tau, particularly when combined with high beta, is a predictor of rapid cognitive decline. So this is where you would hope not to be if you have a PET scan. So people sort of tried to model this in terms of all the things we can measure, the age, the genetics, the measurable accumulation of A-beta and tau, both in the medial temporal lobe and then in the neocortex, leading eventually to synaptic dysfunction, neuronal death, and all of the bad things that happen cognitively as a result of that. So it's just a statistical model of the same concept. So, but this is obviously, far short of a mechanistic understanding, and so we need to get from this kind of model to detailed cellular models of what's driving this process. It nevertheless feels like we have a pretty good handle on what causes the disease. Certainly it's much better than for autism or schizophrenia or other diseases, psychiatric diseases that were discussed this morning. We have strong genetic forms that strongly resemble the more widespread sporadic forms. We have a clear visible pathology. You would think, and the main causes have been known for several decades, you would think this should be a strong candidate for a success story. Unfortunately, as most of us know, that's not the case. I think the best way to describe it would be unmitigated disaster. So the attempts to test new drugs for Alzheimer's, this is surveyed like 244 drugs were tested, entered clinical trials over a 10-year period. The success rate was less than 1%. The only drug that was approved during this period was memantine. It's an NMDA antagonist that apparently is only minimally effective and does nothing to slow down the progression of the disease. So this is hugely disappointing. Things have not gotten any better since that survey, and in particular, we've had a lot of failures of major late-stage clinical trials, so clinical trials targeting the amyloid mechanism. So people have tried to develop, you remember I talked about the two proteases that release the peptide, so beta secretase and gamma secretase, and people have developed inhibitors for both of those, and they have been tested clinically and have all failed so far. In many cases they've been toxic, in other cases they've been tolerated but simply haven't produced any clinical benefit. That's the same story with early attempts to develop a vaccine against the amyloid plaques. The trial had to be stopped because of bad side effects. And then maybe most recently, passive immunization, so the idea of developing antibodies against various forms of amyloid and then infusing them into patients. And so these are all, apart from this last one, which I think is still underway, each of these are monoclonal antibodies, each of which has had a major failure in a late-stage clinical trial. So just to give you a sense of what's involved, I mean, these are thousands of patients, billions of dollars have been spent. I mean, probably collectively, I'm sure tens of billions of dollars. It's enormously expensive. Patients are coming in once a month, in some cases twice a month. They are sitting for 3 or 4 hours in a doctor's office, being infused with these antibodies, massive amounts of antibodies, sometimes a gram per visit. Doing that for 2-3 years, and then you open the code and you find that it has failed. I mean, so this is a real tragedy both for the patients and for, I think, all of us. So something has to change, but that's not so easy. Challenge is it takes a very long time. it costs a huge amount of money, as I already said. It takes a very long time. So the time from when a drug enters clinical development to the time when you actually get the result of a definitive large-scale trial apparently is averaging 13 years in the case of Alzheimer's disease. So what that means is that the failures that we're seeing now reflect the scientific understanding from, in many cases, more than 10 years ago, which you might think that might be an optimistic sign, because maybe the science is better now. Our success rate may go up. We hope so. We talked this morning about animal models. There hasn't been a primate model yet of Alzheimer's, but it would seem like a strong candidate. So hopefully, I know that there are some people who are working to develop one. It's inherently challenging, of course, to model a slowly progressive disease in a mouse that only lives 2 years. But hopefully we will have better models at some point in the future. We also need better biomarkers, and that would include both molecular markers as well as cognitive markers, more sensitive tests for cognitive decline in order to see what's working and what isn't. Importantly, I think we need a way to image not only the mature plaque, which is what the current PET ligand is seeing. We need a way to see the oligomers that are illustrated here. So if these are the guys that are doing the damage, but we can't see them in the living brain. We can detect them in blood. You can detect them in CSF. The blood tests are improving rapidly, and I think that's something that is quite promising. It's very promising for the future, better blood tests, but I think we're not quite there yet in terms of predictive power. Another major problem is that Alzheimer's is frequently comorbid with other pathologies. So people with Alzheimer's, people who have dementia and post-mortem have Alzheimer's pathology, often have other things. They have hippocampal sclerosis. They often have microvascular infarcts. They have TDP-40, a whole slew of other problems. So, you know, even if we develop something that works for Alzheimer's, we may still have to, a clinical trial will have to detect that effect on a background of other problems associated with age. And so just to summarize, the key challenge is we need the right molecular target, we need to develop a drug that hits the right target, and we need to intervene at the right stage of development. I see I'm running low on time, so I don't have a lot of time to tell you what progress we're making here, but I'll summarize very quickly. We need to study the villain, the A-beta that comes from the brains of Alzheimer's patients. You can synthesize the peptide, but it may not be the same as it may not adopt the same conformations as it does in the brain. So we think, my colleagues think a promising strategy is to take extracts from the brains of Alzheimer's patients and test them not just on cultured rodent neurons, but actually on human neurons, which of course we can now do using induced pluripotential stem cells. So you can make stem cells and you can differentiate them very reliably into neurons. You can screen them, you can culture them, you can add these extracts, and this is a time lapse over 3 days. These purple lines represent the growth of new neurites, and you can see that after you add the extract from Alzheimer's brain, you're really inhibiting the growth of new neurites. If you deplete this with an antibody against A-beta, you can rescue that effect, and so you compare. Oops, sorry, I lost. Let's try to get back there. There we go. Compare with depletion. That now gives you an assay. This is a robust, easy assay. And you can test different antibodies, including the antibodies that have been put through clinical trials. So here are some of them here. So the brown line is what happens if you just poison the cells with the Alzheimer's extract. The horizontal line is buffer alone, and then this toxic effect over a 3-day period can be blocked to varying extents by these different antibodies. And so you can use this as a screen to find antibodies that may be more promising. So these, they vary in their effects. The best one is actually a new one that was developed at Brigham, not for a moment claiming that that's going to be the miracle drug to cure Alzheimer's disease, but I think it does illustrate the point that the effects, this is a new assay that can make distinctions that were not previously obvious between these different potentially therapeutic antibodies. So we may be able to do better with assays like this. I'm running quite low on time, so I wanted to quickly talk about the concept of preventative trials. So a major reason for failure, in addition to, major reason for these big clinical failures, in addition to the likelihood that many of them have been hitting the wrong type of amyloid, either mature plaques or oligomeric forms that are not the most toxic form, another very likely problem is that they've been attempting to treat too late. So patients are enrolled after they already have a clinical diagnosis of either mild cognitive impairment or Alzheimer's disease. And if we want to stop the disease, we should intervene earlier. I mean, what we're doing now could be compared to taking a heart attack patient and trying to treat them with statins. You know, statins are great in preventing heart attacks, but they are completely useless once you've had a heart attack. You're not worried about your cholesterol level. You need to get a stent or a TPA or a bypass or something. So we need to intervene earlier. Let's make sure I'm going the correct direction here. So there are a number of attempts underway now to do that. Obviously, if you're going to intervene early, you want to pick a population of patients that you know has a high likelihood of developing the disease within some reasonable period of time. So some studies are studying patients with those highly penetrant familial mutations like presenilin-1, APP. This one is being led out of Washington University in St. Louis. There's another one that's based in Colombia, where there's a large extended family that carry presenilin mutations. And so they're testing, these are antibodies that have been proved to be relatively safe in clinical trials. They haven't worked when applied late, but they may still be successful when given before these patients develop symptoms. And then this trial, which is being led by Risa Sperling at Brigham, we call it the A4 study, the anti-amyloid treatment in asymptomatic AD. And I wanted to just quickly present what they're trying to do here. So this is enrolling clinically normal older individuals who have been screened using that PET test and shown to have high amyloid in their brains. They're going to be treated with one of the big antibody drugs, solanezumab, made by Eli Lilly. So it has failed several big trials, but there were apparently hints in the data that it may be helpful for a subgroup of people. That's not good enough to get it clinically approved, but it's maybe good enough to want to keep trying. So that's the drug, and it was relatively safe, which is one reason why they chose it. So this is a partnership between the National Institute on Aging and Eli Lilly and several philanthropic organizations. This is hugely expensive. This is the budget for this is well over $100 million, and remember that is on top of the what must be several billions of dollars that Lilly has already sunk into the development of solanezumab. So this is not a trivial undertaking. It's going to be enrolled at 67 sites in the US, Australia, Canada, Japan. And conceptually, this is what they're trying to do. So if that's, as I mentioned earlier, if you have a beta in your brain, you're going to have a faster decline than those who don't. The hope of the trial is that we can shift these patients from here to somewhere on that intermediate curve by catching them, at the earliest stage. That's just the geography. But just to give you a sense of what's involved in running a trial like this, this is what you need to do in order to determine whether a drug is working in a disease like Alzheimer's. So they calculated that they needed to look at about 1,100 or so subjects who are positive for PET. So to find those subjects, they had to screen over 15,000 people by phone or web. Those people, like almost 7,000 people, came in for clinical appointments. They're evaluated by a clinical psychologist. They have a whole battery of tests. They're genotyped. They do all these behavioral tests. They then have to undergo a PET scan, so they have to agree to have radioactivity injected into them and go into a PET scanner and also into an MR scanner, because you have to get the structure of their brain as well. And again, I should have said these are cognitively normal people. We're looking for people who are cognitively normal but are at risk for developing cognitive decline. About less than a third of them have elevated amyloid and are enrolled into the trial. And so they, even getting to the start of the trial is a big accomplishment. They got there in December 2017. The trial is going to run for 4.5 years. So the results should come out sometime towards the end of 2022 is when we will know whether this worked. And some of those who are not, who turned out to be negative and are therefore at low risk, are also enrolled in a parallel observational study. And there's also an ethics study to address the question of, what happens when you disclose to patients where, what is their risk? I mean, this is not a trivial thing, right? Somebody in their, typically in their mid-70s, who may or may not be at risk of developing Alzheimer's in their dying years. You want to know, if they're told that they're at high risk, are they going to become depressed or even suicidal? That's an empirical question, and that's going to be addressed in a subset of this study. I won't, in the interest of time, I don't want to go through all of this. They were genotyped for ApoE4, which, as I mentioned, is the major risk factor, the major genetic risk factor for Alzheimer's. And people who carry the E4 allele are more likely to be in the amyloid positive category on the PET scan. So, the point here is that even before we started to test the drug, we can get a lot of useful information just from scanning 4,000 people and correlating their scans with their genotype and their cognitive scores. And we can learn stuff that will be helpful not only for this trial, but for designing the next trial. Because, we can't wait for one trial to be over before designing the next. They have to be staggered if we're going to, get to a cure before that tidal wave hits. This is just so A-beta already is associated with subtle decrease in cognitive score even before it's been detected clinically, using the sensitive behavioral tests that were used in that screening. This is important. As I mentioned, those behavioral tests currently involve, a visit to a professional clinician, the study partner. The patient has to come in with a study partner. It's a fairly big imposition. And if you're trying to do this on, many thousands of people, they've now developed a 15-question questionnaire, which actually turns out to be surprisingly good at enriching, at least, for people who have amyloid deposits. So questions like, have you experienced memory decline? Have you seen a doctor about your memory? Do you have trouble misplacing things? And so forth. Without going through the data, a key point here is that it's sometimes supposed that patients wouldn't be aware. If you're demented, you won't know it, because if you knew it, you couldn't be demented. The idea that they would have anosognosia. That turns out not to be true, at least based on this evidence. So people who, in their self-reports, express more concerns about their cognition are indeed more likely to have amyloid deposit. It's a pretty good correlation with their partner's score, and you can get an even better enrichment if you combine both of these things. So that's maybe not scientifically interesting, but it's practically important, if the difference between a day of professional clinical evaluation versus a 15-point questionnaire that can be delivered over the phone. I won't go through this. This just says that, as with amyloid, also with tau, you see the tau pathology, and people who have more of this, on average, have both higher amyloid, and sorry, I should have said it the other way around. People with higher amyloid have more tau, and also have less good cognitive scores. So I think the take-home, the big point from this is that this combination of screening, brain imaging, behavioral screening, and genetics is already giving us useful epidemiological information that, for future studies. So this is the hope. I don't have time, but I will say that the next study, this one is going to be done in 2024. The next one is already underway, and the idea is to enroll people at an even earlier age, 55, starting at 55 rather than 65. They haven't started that yet. They're just still choosing the therapeutic agent, and so that's a big negotiation with the drug company to determine what agent to use. But hopefully we'll be able to report back on that at some point before too long. So it's all very tough, right? It all seems rather alarming. I think, so my colleagues like to point to success stories from other fields, such as heart disease. Remember how long it took to develop cholesterol therapies and to prove that they could save lives. It's probably the same with Alzheimer's. It will take time, but we are hopeful that we will get there in the end. I actually took this photo from my office window at Brigham, so you see the storm clouds on the horizon, but we also see the rainbow. I think that's certainly the attitude of my colleagues, who I would like to acknowledge here: Dennis, Risa, Keith Johnson, Tracy, Christina Leiliu, Dominic Walsh, many, many other people who've contributed to this. So thank you very much.
We have bit time for a few questions. Yes.
Yeah. Thank you for the wonderful talk. I'm here.
Yeah.
Okay. I just wanted to point out to the fact that even patients with the same level of brain burden, same AD patient,
same level of
brain burden, like damage, can have really different level of cognitive decline, cognitive impairment, right? Recently, it was proposed to use a measure for this discrepancy called the cognitive reserve to account for this buffer. So, what do you think? Do you think that including a measure of cognitive reserve in the diagnosis of AD or even in the prognosis of AD will in some way improve the capacity of...
I get the question. It might. If you note this section, so with the pointer, this might sort of partly address your question. So there was no difference when they compared the people who have high A-beta with those who don't. There was really no significant difference in the amount of education. So although it has been widely claimed that education is protective, which would presumably be one source of cognitive reserve, that at least doesn't seem to be true here in terms of amyloid burden. You're probably asking a related question, which is it protective against the development of clinical symptoms?
Yeah.
I mean, I think so. I don't know the data on that. If you start out with more, you're undoubtedly going to perform better. I mean, that is why you need to do longitudinal tests. You need to measure the same subject repeatedly. It's much more powerful than cross-sectional designs. Yeah.
Thank you.
So could it be that these 244 trials failed because A-beta is not really the primary culprit, that this is just an epiphenomenon of a completely different molecular mechanism?
No, because they're not, those 244 failures, they are not all amyloid trials, and many other things have been tried. I think I've emphasized the amyloid trials partly because I think that's the strongest hypothesis in town. It's the one that has been most thoroughly explored. It's the one into which most money has been invested, that industry has placed the most hope. I do not want to suggest that that's the only possible mechanism. There are certainly trials underway. I didn't have time to mention it. There are trials underway now to target tau. There are trials targeting the TREM2. And there are certainly other mechanisms. I think it's from the genetic evidence. It's in my mind almost inconceivable that this is not important in some way. You cannot get around the fact that all these mutations affect amyloid processing. But undoubtedly, there's something else happening. Amyloid problems cause many other things. And amyloid, particularly if those problems begin early, they may not be the best place to intervene. If we knew what happens subsequently, we could target that.
The question was a little bit different.
Okay, sorry.
Could it not be that alpha beta in tau are the end points of disease mechanisms, and to target endpoints is useless? Could this possibility
Well, I think the genetic evidence suggests that they are causal. The fact that there are highly penetrant mutations in both the Alzheimer's precursor protein and in the protease that releases the A-beta peptide strongly suggests that this peptide is important. You know, if you want more proof, I mean, you know, anybody would welcome more experiments to nail it further. As I say, it's not the only thing that's happening, but it does seem it's the one thing that we know that clearly has a causal role. So, yeah. Yeah.
Yeah.
Bob.
Go ahead.
So, with the failures of the amyloid tests, doesn't that raise questions whether the amyloid hypothesis is a good one, or has it basically just shifted the question to, well, it's a good hypothesis, but we have to get there earlier?
I think both things go. Well, certainly, I still think it's a good hypothesis. It's not a very precisely specified hypothesis. I mean, maybe it may have been a disservice to call it the amyloid hypothesis as if it was just one thing. Maybe the universe of possible explanations for why, you know, these genetic forms all seem to converge, or mostly converge, on amyloid. So, you know, there's clearly a target there that, in principle, ought to be targetable. But you know, just to repeat, I'm not advocating that we should only pursue that. I mean, other things should be pursued and will be pursued. Yeah.
Thank you, Dr. Jennings, for this very clear overview. You showed that the tau spreads sequentially. I want to point to the fact that the tau pathology actually starts much earlier than had originally been thought. There was a neuroanatomist who showed that at the age of 10 to 30 years, so that gives us even more time to develop new procedures.
Right.
You can find in the brainstem the first subtle pathologies of tau.
Right, although whether all of those people will then go on to develop disease, I don't know.
Certainly a tricky point, but yeah, you know, if that's the tau that spreads further, and I think there's an anatomical evidence, then this is probably, and it's probably a neuromodulatory problem, because it's the nuclei that are affected first.
Yeah, could be. I should have mentioned there's no genetic evidence that I know to directly connect tau to the causal pathway. So there are mutations in tau that cause other neurodegenerative diseases, but Alzheimer's is not one of them. So we don't know the reason for that. But I think everybody in the field thinks that tau you know, it's not just, nobody thinks it's purely epiphenomenal. There's actually a bunch of cell biological evidence that it is functionally important.
There's one final question, please.
Yeah, sure.
All these drug failures do remind me of brain cancer. So I would like to raise the question: are the drugs really entering the brain? Because there's a blood-brain barrier, as we all know. Do the drugs really enter the brain, or are they just swimming around in blood?
I'm not sure I quite caught the question. Your question was, do these drugs enter the brain? So, yes, they do, but not very much. And that's actually one very plausible reason for the difficulty in treatment, is that you infuse, and it also accounts for the huge cost of treatment. If you're infusing it intravenously, only a small fraction of the antibody gets into the brain. I'm not sure if it's known how it gets in. The majority of it just circulates in the blood and is basically money down the tube. So yeah, that's a serious issue. But you know, developing antibodies that cross the blood-brain barrier is a big protein engineering challenge. People are working on that, but we're not there yet.
Okay, thank you. We have to move on to the last talk.